agarose gel. Now, this cell-free DNA, basically a library preparation is done. So basically, the isolated cell-free DNA is first purified, then it is enriched, and then we go on and do the sequencing. Basically, what is done is that the sequencing that is read is compared to a reference genome, which was GRCH38 that was used in our study. And after that, once you've identified the sequences, you look for the variants and decide whether they are pathogenic or not. So Varimat pipeline was used for the annotation. Now, we did this in 26 eyes of 25 patients. They were mainly male patients with bilaterality of disease in 7 patients. 20 of the eyes underwent enucleation. In 6 eyes, we were giving intravitreal chemotherapy, so they were undergoing salvage treatment. And we were able to get cell-free DNA in all the cases. And this was cell-free DNA because by the characteristic of cell-free DNA is that it peaks at 140 to 160 base pair. That's the size of the base pairs that you get for these exomes. Now, in 25 eyes, the mean concentration that we found for the cell-free DNA was 23.65. It was higher for patients that underwent enucleation compared to the ones where we were doing globe salvage treatment. So we were giving intravitreal chemotherapy. This is the data sheet showing all the cases that we did. Now, essentially, we could do targeted exome sequencing only in 4 samples of aqueous and 2 tumor tissues, basically because of financial constraints. And this is what we found. So in 2 patients where we did both tumor tissue and aqueous, we did find some correlation, or we find similar pathogenic nonsense. Can I continue? This is just the last slide. Yes. So yes, so here in aqueous humor and tumor tissue, the same mutation was found, which was nonsense and pathogenic. In the other case, we did find RB1 gene mutations, but they were not similar for tumor tissue and aqueous humor. But we were able to find mutations in all aqueous humor samples, and all of them were pathogenic for retinoblastoma. So I'll skip the discussion and I'll just go on to what finally I want to say here is that our study validates the previous reports that aqueous humor may be used as a source of tumor-derived cell-free DNA. This is a preliminary study and the first one from India which has reproduced the same results that have been done in the West, and we need to do it more to be able to reproduce in rest of the centers. But it's a big step forward in prognostication for globe salvage and not just survival in these patients. Thank you. Thank you.
Good. It's more of a laboratory-based thing.
Yes, ma'am.
And what is its clinical implication?
It has a huge clinical implication, ma'am, in the sense that now intravitreal chemotherapy is being used extensively in advanced intraocular retinoblastoma for globe salvage. So if we have diffuse vitreous seeds, we give intravitreal chemotherapy, which was not being done a decade ago. So we are anyways putting the needle inside. So what we do is, before we do intravitreal chemotherapy, the aqueous sample is taken to reduce the intraocular pressure. Paracentesis, as it is a part of the procedure in many centers. So we had the aqueous sample. Now this sample can actually be used to prognosticate whether you will be able to salvage that eye or not based on the mutations you identify.
What is the cost incurred for this?
Yeah, it is very costly actually. So it is around 30,000 for a single sample exome sequencing. That's why we could do it only in 4 patients. But we now have a collaborative project on the same, and we want to do more work on this.
So 4 patients is too little.
Yes. It is definitely costly, ma'am, but if it finds its way, it will have a huge implication on the globe salvage.
Let's see.
Right. Okay. Thank you.